Welcome to today's lecture. Today actually what we are going to study is different tools and techniques. Okay. Tools and techniques used in study immunology, or the different techniques which is also known as immunotechniques, that we will discuss today. This lecture actually we thought to give today for two main reasons. One is the tools and techniques that we are going to discuss, that will help to understand the many experiments that we are going to discuss in future classes. So, you can understand better if you know what are the techniques. If I tell that just the name, okay, immunolocalization, you may not understand what is that mean or what is the outcome of that. So, that is why I decided to discuss few techniques to understand the immune system better, number one, and number two, I am sure by now so many days just you are listening T cell, B cell, MHC1, MHC2, T cell receptor, B cell receptor, macrophage, dendritic cells. So, you kind of monotonous lecture one after another, that continuously we are discussing. Definitely we are, that is our main goal, but before that if we understand few techniques or how this antigen antibody we determine. If you remember, like the primary response as negative response, we showed you a graph, right. So, at the very beginning there was no antibody and then it is increasing. So, how this graph is made? Because somebody need to measure the amount of antibody, how much antibody is there in the serum. Or if you remember the table where we showed that what is the amount of different isotypes of antibody in serum, like how much milligram is there. So, how we get that information? So, to understand that, so we need to know few techniques. So, today what we are going to do, we are going to discuss some tools and techniques that we normally use to study immunology. And one more important thing is that the techniques that we are going to discuss in next 4-5 lectures, different techniques, this is not restricted to immune system only, because immunotechniques are very popular. I mean, you believe me that very popular to study any almost most of the branch of biology or life science, like microbiology, molecular biology, proteins, cell biology, these are used everywhere. Okay. Because using these techniques we can understand many things that you learn with time, and this is one important thing, that these techniques are used almost every branches of life science. And second is, these techniques most of the time is very cheap. Okay. You do not need too hi-fi instrument or equipments or costly equipments. You do not need much expertise, but definitely you have to do the experiment by yourself to know the tidbits, but we are not going to go detail about the technique. Mostly what we are going to do is try to understand the principle of the techniques or tools, and what information we can get and how it looks like, whether it is a number or it is a photograph or it is something else. So, these we are going to discuss. Okay. So, the first definition wise, immunotechniques are, you can say, any techniques which use antibody for one of the reagents, then it is we call immunotechniques. So, any techniques we use antibody as one of the major reagents or most important reagents, we call it immunotechniques. Okay. So, let us start. So, today's it's lecture 26, it is a tool and techniques. Okay. So, first what we are going to do is, we are going to discuss immunotechniques, and one of the major reagents of these techniques should be antibody. Okay. You know that antibody we write like AB in short, and antigen we write AG in short. So, antibody and antigen we are going to use AB and AG very frequently. Clear? So, now the first thing, if you would like to find antibody, what you need? You need antigen. you need antigen, and if you are studying any particular antigen specifically, first thing what we should have in our hand is pure antigen. Okay. We should have pure antigen, and most of the time what is this pure antigen? Pure antigen is, that means you have to have a pure protein in your hand. Okay. So, pure protein means you have to purify the protein. That source of protein may be natural source, like from, say, if you want to purify protein from cell, you can extract that total protein from cell and purify your protein of interest. I am not going to go all these details how you will get the protein, because it is a lot of class you need to discuss. Just, or you can express the protein in recombinant way, right, and then purify your recombinant protein of your protein of interest. Like you clone the gene, express the protein, you can express the protein in bacteria, you can express the protein in yeast, baculovirus system, animal cell, plant cell, whatever the original source of the protein, whether it is a recombinant or native source, you have to purify the protein. Protein purification, there are several techniques. You can go for affinity purification, you can ion exchange chromatography, and then you can purify by immunoprecipitation. That we will come and discuss what is this immunoprecipitation is. So, then your protein is your hand, and you have to raise the antibody. Okay. So, normally what we do to raise the antibody, because one of the reagent of immunotechnics is antigen. If you are looking for antibody or measure antibody, detect antibody, then you need pure antigen in your hand, that is your bait. So, antibody will bind and you can measure that, the antibody part. Or if you would like to characterize the antigen or purify the antigen or do some localization of the antigen, so then what you have to do? You need antibody in your hand. Okay. So, it is general. So, if you need to characterize or know about the antigen, you need antibody. If you want to know anything about the antibody, then you need antigen. So, antigen you have to have by purification, expression by in recombinant system. There are several methods, right. So, I am not going, assume that I have pure antigen in my hand. So, if I have pure antigen in my hand, what I have to do to raise antibody? Normal practice is we inject that antigen into animal. Okay. So, you can use the mouse, you can use rabbit, you can use goat, monkey, horse, depending on how much antigen antibody you want. If your requirement is very little, you can use small animal. If your requirement is very high, you can use big animal. And if you use big animal, there is one more factor is there. If you use big animal, then you need more amount of pure antigen, right. So, antigen purification or pure protein in your hand, pure means pure to homogeneity, that means no other protein contaminant should be there, because if you inject 2 protein into animal, antibody will be raised against 2 proteins, but if I would like to work with 1 protein, that should be very, very pure. Okay. That should be in our hand. So, pure protein means pure to homogeneity, nothing else is there. So, if you want to use big animal, you have to have good amount of pure protein, and that is not always easy, because having pure protein in good or large amount is one of the key factor to raise antibody. So, in laboratory purpose, what we do is, most of the time, if it is not a commercial requirement, in laboratory purpose we do not need that much. Okay. So, mostly we use rabbit is fine, okay, or mouse. So, you have pure antigen in your hand. So, pure antigen you have to inject into animal. Okay. So, you have to inject into animal, and so that injection what we will do, that injection, that antigen will you have to administer into animal different way. Many of you know, I am not going to discuss about detail about it, why and when, but there are many ways you can administer. You can do intramuscular, intravenous, intraperitoneal, subcutaneous, oral. Okay. So, normally we do intramuscular in laboratory practice for raising antibody, and that we will discuss little more detail about this in vaccine production or during that. Okay, what is vaccine, how you can produce or how you can therapy. So, if you inject the antigen, and what will happen, you remember this, I am sure that you remember this graph. Okay, so, initially few days you will not see anything, then it will increase, then go come down, then what we will do is to raise more and good quality antibody, we will inject again antibody in the same animal twice. Say maybe after 3 weeks of first injection, we will do the first booster, and what will happen? You know that it will reach maximum good amount, good quality and IgG type, right. So, this will continue. So, if you need more, again you inject again, because it is already primed. Okay. At this stage, so when this first booster dose, what we do is we isolate or take some blood. Okay. We isolate the blood, and from that blood we purify the serum. Okay. We purify serum. How you get the serum? You know difference between serum and plasma, right? I am sure you know. So, plasma is, I am repeating again, plasma is if you isolate the blood, just centrifuge immediately in presence of say anticoagulant, what will happen? All the blood cells like WBC, RBC, all these will precipitate, and then you will find a liquid part or fluid part as supernatant. So, that supernatant still contains the clotting factors. So, what will happen if you keep that fluid, it will clot. So, this is plasma, but if I isolate the blood, and after isolation of the blood, if you allow them to stay, say normally what we do is we keep it 37 degrees centigrade say for 1 hour. Okay. After that what will happen? Most of the blood will clot, right, and even after clotting some fluid will still there, that is actually serum. So, after 37 we keep it 4 degree overnight, 4 degrees centigrade overnight, okay, and then next day we just collect the centrifuge the cells and slowly without disturbing much, because if you disturb it then RBC will lyse and it will take a unnecessary red color. You can avoid that by handling gently. So, that 4 degree then you centrifuge and collect the fluid, that is serum. So, what happened, all the clotting factor you know that that will be separated by that. So, these serum actually contain, this serum contains the antibody, and this antibody we can purify, and this antibody we can use directly, the serum can be used for different techniques, or in some cases where we need very good quality antibody, or some techniques need pure antibody. So, because serum contains what? Serum, any serum you take from say bovine, rabbit, human, you know the major constituents of the serum is what? Major constitution of the serum is the albumin, serum albumin. So, if there are too much serum albumin, that may mask the antibody molecule. So, many times what we do, we purify the antibody from that serum. Okay. So, I am not going detail here at this moment, but we can purify the antibody just by affinity chromatography. Those who are interested, go and read that affinity purification of antibody using protein A. Okay. Protein A affinity purification is normally used to purify IgG. Okay. So, that way what you can do, from the serum only IgG you can separate. So, now after this thing is done, so it will take time, because you need a pure protein, then inject, you have to wait 2 to 3 months, and then after that you isolate the draw the blood, then you isolate the serum and you test, because we are not going to kill the animal. Okay, animal will still survive, we are not killing or sacrificing the animal to get all the blood. So, keeping the animal alive or alive, we isolate some or draw some amount of blood, from there we isolate serum, and that serum will contain antibody. Antibody against what? Antibody against of that pure antigen that you injected, protein of interest. That antibody you can purify using protein A. So, now this is your reagent to study. Okay. Now, this is your reagent to study. So, what we will do with this? So, now if I say that okay, this antigen you purified is not good, so that is not going to work better. So, how do you check what is this antibody is good or bad, or is at all antibody is produced or not? Because many times what happen, your antigen may not be good. Antigen may not be good means it may not have any B cell epitope. If there is no B cell epitope, no antibody. If there is no T cell epitope, no antibody. So, normally it is very rare cases that there is no B cell epitope or no T cell epitope if the protein. Generally, rule of thumb is if the protein molecular weight is more than 5 kilodalton, normally it is act as immunogen. You remember, I hope that you remember what is antigen and immunogen. Okay, I am repeating again. Antigen is the molecule to which antibody can bind. It can be anything, antibody can bind is antigen. But if antigen you inject into animal, if it can elicit immune response, that is called immunogen. That means, any antigen can elicit immune response is antigen, is immunogen. That means what? All immunogens are antigen, but some antigen may not be immunogen. That means, antibody can bind, but it cannot elicit immune response. So, in that kind of thing, non-immunogenic antigen, if you inject into animal, then it will not raise any antibody, but you can check that normally it is not that common. So, normally most of the proteins are immunogenic. So, they are good enough to raise the antibody in animal, but still we have to check how good the antibody is, right. So, what we do is the one of the very simple test is immunodiffusion. Very simple test is immunodiffusion. What we do there? In immunodiffusion there are single immunodiffusion or double immunodiffusion. Okay, two things is possible. Okay. So, for that what we will explain is for immunoprecipitation immunodiffusion, that means what is happening in this here in immunodiffusion. I will show this picture, I will just listen to me first. So, I will explain this, whatever you are seeing I will explain this why this is here. So, in immunodiffusion what we see is if antigen and antibody binds or react, okay, they will precipitate, and we can see a very nice precipitation line or precipitin line. Okay. So, if you see any precipitation, that means antibody is there. Okay. So, what is happening? You can titrate also agglutination or blood group test, it is just the precipitation or agglutination kind of thing. So, what we can do is, suppose this is I am seeing a graph, assuming that we can see the precipitation we can see. Now, in a tube, okay, in a tube you take a solution where lots of antibody is there. Okay. This is antibody solution. So, huge amount of antibody is there. Okay. Then you are drop wise you are adding antigen. Okay. So, initially what happened? Initially your concentration of antigen, if this is the antigen concentration, you are adding. So, amount of antibody is fixed, you are drop wise adding antigen. So, initially antigen concentration at the very beginning it is 0, and then gradually it is increasing. Okay. It is increasing. So, if I draw this way, it is the amount is increasing in amount, I mean the antigen concentration is increasing. So, what we will see is initially there is no precipitation, at the middle there will be a precipitation. So, this is the amount of precipitation I am drawing. Okay. There you will see the precipitation, again it will not. So, what we will see, if you keep on adding, solution is clear, 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 and suddenly you see some cloudy precipitation is happening, and then cloudiness is increasing. Then if you keep on adding, then again that cloudiness will be cleared. Okay. And this region, that where you are going to see the cloudiness, how you can measure that cloudiness? You can measure the cloudiness by simple spectrophotometer. So, if I measure by OD, okay, so the turbidity will increase, and if the turbidity increases, you can see that OD is increasing. So, OD will increase, then again going down back to the minimum one, right. This region where you see the precipitation or you see the cloudiness is called zone of equivalence. Zone of equivalence. Why initially there was no precipitation? Initial, what was the condition? You know that I already told that antigen antibody interaction is not a covalent interaction, it is a non-covalent interaction. So, there is depending on the affinity, they bind and open, bind and open, it is a continuous process. If a high affinity, after binding it will remain for a long time. If it is low affinity, it will bind, after some time it will open, and it will find another molecule of antigen, it will bind there, open, another bind there. So, it is they will continuously exchange, and there will be an equilibrium. So, at the early stage when there are huge amount of antibody, so antibody amount is very high, what will happen? When the antibody amount is very high, condition is assumed like this. That means, too many antibody molecule, only few, this red are antigen and blue is the antibody. Okay. So, too many antibodies, but very few antigen molecules are there. So, they will make like this. So, individually they will find. So, there will be no, because to precipitate something, it should be little bit heavier or make a bigger complex, so that it pull down. Okay. So, as long as this is there, you will not see any, I mean as long as this is there, okay. So, that time you will not see any precipitation. But if you keep on adding, there will be a time where they will find this or this kind of structure. So, it is just the ratio of antigen and antibody is such. So, they will make a nice kind of structure or bigger structure. So, 4 antibody, 4 antigen, 3 antibody, 3 antigen, they will make bigger structure. You might have seen much better picture in when I was showing you the slide, same slide, I mean similar structure. So, when they make this big structure, then they cannot stay in the solution anymore, they will precipitate. Clear? Now, if this region or this concentration or this ratio will reach at certain point. So, maybe this is the region where this kind of structure are formed. Again, if you keep on continuing the antigen. So, you saw the antibody and keep on using keep on adding the antigen, what will happen? In that case, this is the scenario where antigen number is much more than your antibody. Again, what will happen? Antibody will be diluted and you will not have this kind of structure. If there is not this kind of structure, or this kind of structure will break and that will be diluted. So, you will have this kind of distribution, you lose the precipitation. So, at a particular dilution or particular ratio of antigen and antibody will form this kind of structure, only then it will precipitate. This is whatever I am talking, this is whatever I am discussing right now, it is I am talking about the antigen antibody reaction in solution. But here you cannot see the precipitation line, right. So, that is why the technique immunoprecipitation that we are using. Okay. So, in immunodiffusion, what is happening? There are single or radial immunodiffusion and another is double immunodiffusion. Single immunodiffusion, what we do is, all of you know the agar plate. Okay. So, you make a 2% agar solution and put on a slide or in a Petri plate. Okay. So, you have a glass plate. So, assume and then you put some 2% or 1.8% agar. Okay. Normally, we are using agarose, okay, not agar, that we are using in bacteria. No, 2% agarose solution we add. So, it is the in one slide. So, it is not one dimensional, or what we can do is we can use the Petri plate. I am sure all of you know what is Petri plate is, where you grow the bacteria. In that Petri plate you pour the agar. How thick? Maybe say 3 to 4 millimeter thickness, and then you make hole, okay, different holes. Just punch a glass pipe or plastic pipette kind of thing and make a hole or well kind of thing. So, while you are pouring this agarose, what you can do is you can mix your antibody. So, that means what happened? Your antibody is everywhere. Your antibody is everywhere. Okay. So, if this is a say plate, suppose this is the antibody is everywhere. Okay. This is your antibody, it is everywhere. You cannot see that, definitely you cannot see that, okay, because it is not visible, but in this hole, okay, in this hole or well, now you add antigen, okay, antigen solution, that pure antigen solution, pure antigen you have, you pour antigen. So, what will happen if there is, now I am drawing this again like this, and there is a hole here, antibody is everywhere, equal concentration, and you are putting antigen here, and this antigen, what will happen with time? It will diffuse, right? It will diffuse, and it will diffuse sidewise, I mean according to radiation it will diffuse. So, while it will diffuse in different direction, okay, antigen has a particular concentration, okay, while it will diffuse. So, at the very close to the well, suppose this is a well full of antigen, very close to the well the antigen concentration is very high, and more it go, it will be diluted. So, at a particular concentration, while it is diluting, at a particular concentration what will happen? It will reach that equivalence, zone of equivalence. That means, the ratio will be perfect to make precipitate, and will be perfect to make precipitate. Then what we will see? We will see, I mean this is the thing. So, what we will see? This is a nice circular Petri plate. Okay. This is a nice circular Petri plate. Let me take this little this side. This is a nice circular Petri plate, and you have a well which you won't see anything, because all neither antigen nor antibody is color. So, what we will see, and the next day if you keep this plate overnight at 15 degree centigrade. Why you keep in 15 degree centigrade? Diffusion rate is high in a high temperature. We keep it 15 degree centigrade because less chance of contamination, because bacteria may grow in the agar. So, that we would like to avoid that, and diffusion will be little slower. So, chance of formation of precipitate is more. So, what will happen? You will see a very, I am drawing the yellow, it is actually is very faint milky white. So, you will see very nice ring, okay. You will see a very nice ring. So, next morning you come, you see against the light, you take the light, against the light you see a very nice faint milky white ring. If ring is there, that means antibody is there, because what you did, you isolate the serum, mix with the agarose and plate it, and you know antigen is there, because you have purified antigen, you use it before, but antibody is there or not, you did not know. If you see the precipitation line, then or a very nice circle, that means antibody is there. So, if this is the case, what is the advantage? You do not need any instrument except one Petri plate and little bit agarose. You can tell whether antibody is there or not. Okay. So, and these thing you cannot, I mean you can tell that how, what is the amount is of antibody is good or bad, but normally we do not use this, we use double diffusion. What is there in the double diffusion? We in double diffusion, same Petri plate. Okay, let me use this. So, in the same Petri plate, what we do is we make multiple hole. Okay. Multiple hole means there is one hole in the center, then radially we will put make different hole. In better is uniform distance is better if you can punch it. Okay. In the center hole we add antibody, and in different hole you can do either way. Okay. In the center, suppose in the center you add antigen, either way is possible. Okay. So, in center you add antibody, and in different hole you use antigen of different concentration. Suppose this is x, this is 2x amount, then 3x amount, and then 4x amount, and then 5x amount. This amount, x amount, 2x amount, whatever, maybe 2 microgram, 1 microgram, that you can measure. So, and you do the same thing, what will happen? Antibody will diffuse all direction equally. Same way, if the antigen color if I make red, okay, that will also diffuse in all direction, in all from all well, right. So, what will happen? For each case there will be a precipitation line, right. So, if you have less amount of antigen, it will dilute it very quickly. If you have more amount of antigen, it will be diluted, I mean in comparison to x, 5x, what will happen? The dilution will be much less in comparison to x. So, if I now draw, or if I now make one well here where we put antibody here and one well here antigen, another well antigen, same amount if I give, what we will see? At a particular distance from this well, we will see a precipitation line. Same distance I will see a precipitation line. So, next morning I will see a nice faint milky white or good milky white precipitation line here. So, if the amount of antigen, if this is antigen and here is antibody are same. So, say suppose 2 microgram, 2 microgram, serum I do not know. Okay. And if I give double amount of antigen here, what will happen? Double amount of antigen means this will be if this is say 2x amount, this is x amount. Where it will form? Because if this is x, if this is the proper dilution, that dilution will reach little far, okay, because it will dilute more. So, instead of like we will get this, it will be closer to this antibody well. Okay. Same way we can just do, in center we can keep antigen, and the serum we isolate, we gave say x amount, we gave 2x amount, then 3x amount, then 4x amount, or we cannot increase that. What we will do? This is undiluted, okay. Then you diluted 0.2 times, then 0.3, 0.4, you dilute. So, 1 is directly the serum, then you direct half. So, let me put this way, half, then then 1/4 x, then 1/8 x. What will happen? More you diluting the antibody, the amount of antibody is less. okay, and here suppose you give 1/16th dilution of serum. So, X will give you better precipitation line, and gradually the antibody is diluted, you will see the line are getting faint and faint, and 1/16th you may not get anything. So, what this will tell? This will tell, suppose you get a very faint precipitation line even in 1/50th, 1/50 dilution is also giving you precipitation line. That means what? That means your antibody amount in the serum is very good, because even you diluted 50 times, still you are getting precipitation line. Okay. So, this way double diffusion can give you an idea how good your antibody amount, and definitely whether antibody is produced or not, you can figure it out, right. This is called Ochterlony double diffusion method, Ochterlony double diffusion method. So, you just type in the net immunodiffusion and search for picture, you will get variety of picture, variety of this kind of drawing much better, because this is drawn on scale and compass and using, and there are some real life picture, and you have an idea what is immunodiffusion is. Okay. So, thank you for today or this lecture. In next lecture we are going to discuss another technique. Till then, bye.